Hello, I'm John Gribben from the Barts Cancer Institute in London, and welcome to the 20th Annual International Workshop in Non-Hodgkin's Lymphoma here in Scottsdale, Arizona. We just had our first session focusing on the microenvironment in lymphoma, and I'm joined by my good friend and colleague, Randy Gascoyne from the British Columbia Cancer Agency.
Morning, John.
So, Randy, we've just had a discussion on the microenvironment, and here the first session of the whole meeting focusing on the microenvironment. For a long time, of course, we focused on the lymphoma naturally in this disease, but we've long recognized that the lymphoma cells don't exist there in isolation, and a lot of work now looking at those just in terms of understanding how they play a role both in the pathogenesis in the disease, but also more recently how they may also serve as a therapeutic target. So what originally got you interested in focusing on the microenvironment?
Well, very good question. To some extent, as you mentioned, our colleagues in the epithelial malignancies and melanoma have had a longstanding interest in the microenvironment, and curiously, those of us in the lymphoid field largely ignored it, as you said, because we were focused on the tumor cells, which themselves are lymphoid cells. To be honest, I got interested in it because when one looked at the spectrum of microenvironments across the spectrum of lymphoid tumors, it's quite profound. So everything from Hodgkin lymphoma, where you have tumor cells that are there at 1%, and actually the bulk of the biopsy is microenvironment, it was sort of an obvious clue that there must be gold in being able to actually study the microenvironment. But the same is true across all lymphomas, primary mediastinal, follicular lymphoma, DLBCL. They all have a profound component of microenvironment. And in some instances, it's there to provide growth and survival signals to the malignant cells. In other instances, it's all about immune evasion, actually, which is very prominent in a number of tumors.
So when you think about the immune environment and you think about the dichotomy between signals that can provide growth to the B cell, in fact, the role of T cell B cell interactions in normal lymphoid biology is probably what's being recapitulated to some extent. Yeah. But there should also be an attempt by the body to reject the tumor. And what's unique about the lymphoma microenvironment that subverts that approach?
Yeah, so for some of the lymphoid cancers, we don't have a full understanding of that. We know that those cells are there. We know that they're armed. Some of your own data has very nicely showed that actually there are issues to do with the synapse formation in follicular lymphoma that can be overcome by some medications. In other instances, we know that the tumor is proactive and actually has a dominant oncogenic effect of actually evading immune attack. And for example, some of our own work where I think it's been very interesting is diffuse large B-cell lymphoma, where we now know there are a number of mutations that clearly affect genes that have to do with the crosstalk between the microenvironment and the tumor. And the tumor does certain things to evade immune attack. And in general terms, they downregulate certain molecules on the surface, such as HLA class 1, class 2, etc., to be able to hide from the immune system. But at the same time, they also upregulate other molecules that actually fend off the actual attack by the immune system. And some candidates would be in large cell lymphoma, for instance, beta-2 microglobulin gets downregulated, CD58 gets downregulated. The tumor cells upregulate PD-1 in some settings, or pardon me, the ligands of PD-1, not PD-1 itself. And so a number of strategies the tumors use. And I think the genomic data at the time we are right now in 2014 is very exciting, because now we have absolutely credible data that in fact the tumors use that as a prominent mechanism to avoid being attacked by the immune system. And as you mentioned, for years we largely ignored that whole component of the biology of lymphoid cancers, and now it's front and center.
So there's two components you talked about there that I want to follow up on. The first one is clearly, I think it's now accepted that the microenvironment is playing an important role in the prognosis of the disease. But these are, after all, genetic driven processes, and we know that it's the genetic of the cancer that largely drives the prognosis. Yep. In what way do you think the genetics of a cancer can help to drive a different microenvironment in an aggressive lymphoma compared to potentially in a less aggressive lymphoma that looks looks down the microscope to your eyes the same?
Yeah, so I think if you were to contrast follicular lymphoma, for instance, and diffuse large B-cell lymphoma, I think in follicular lymphoma, the dominant things going on in terms of the interaction between the tumor cells and the microenvironment are more about growth and survival signals. And it learns to coexist with the microenvironment there and fend off attack. But I think those are more subtle mechanisms the tumors use to do that. I'm not convinced at this point that we have good mutational data, that it's actually somatically acquired mutations that lead to that actual coexistence. In contrast, in large cell lymphoma, we know there are 8 to 10 genes that have a direct impact on immune cells in the microenvironment. And by altering those genes, either through mutation and or copy number alterations or even translocations, the tumor can then coexist with those immune cells and it becomes a dominant oncogenic mechanism. I think the data are much more convincing in diffuse large B-cell lymphoma and in primary mediastinal large, but I think there are also compelling data now beginning to emerge in Hodgkin lymphoma that the tumor does the same thing.